Hello, everyone. What we're going to do in this lecture, we're going to go through an overview of how you can actually clone a DNA molecule into a specific vector type, which are plasmid. So, what I'm planning to talk about here is first of all, we'll go through an overview of the whole process. How do you go from planning the experiment all the way to actually isolate the DNA that will contain the sequence you want to clone? Then we'll talk quickly about the type of vectors that you want to use, and then we'll go through step by step. So how can you produce what we call the insert? You can produce it by PCR or by doing restriction enzyme. We'll focus mostly on restriction enzyme cloning. And then we'll go through an example of how you can plan which restriction enzymes to use to actually cut out the insert and then stick it back into or stick it into a new plasmid. Then we'll talk about briefly the process of ligation, transformation, and selection. So how you isolate those bacteria ultimately that do contain the recombinant plasmid containing that sequence that you want to clone, and so how you do the analysis at the end. So first of all, a few words about cloning. Cloning is a generic word that simply means to create identical copies of something. If you think you may be cloning a CD, sorry, or you can do a clone of different things. When it comes to living organisms, it means creating genetic identical copies, so individually genetically identical of different type of living organism. Could be viruses, could be cells, and so on. And when it comes to DNA, what we're talking about is making millions or billions of copies of identical molecules of DNA. Identical molecule of DNAs that can be of different types. It could be a copy of an mRNA, of a mature mRNA, so without introns, which is referred to as a cDNA, or it could be just a copy of a promoter, of one part of the promoter, one part of a protein with a certain part of our open reading frame. It can be anything. But if you're making millions of copies of that sequence of DNA, you're making a clone. Now, what are the applications? There are many applications of them, right? You can be trying to clone, for example, a cDNA, so a copy of an mRNA, to make large quantities of a protein. That's how insulin or growth hormones are made in the lab and then purified to be actually sold on the market. You can do it because you want to study the function of a protein. I've done it during my PhD, for example. We cloned calreticulin, which is a protein that is highly expressed in the endoplasmic reticulum. It's found in other parts of the cell as well, but highly expressed in the endoplasmic reticulum. And we show that by expressing a lot of this protein in cells, you could actually increase the amount of calcium stored in the endoplasmic reticulum, showing that that protein indeed is important for that function. You can do cloning to study the mechanism of the expression of genes. You can clone a promoter or an enhancer, which is a sequence that do increase the activity of promoters. We've done it in my postdoc to, for example, to study the importance of one sequence close to the Duchenne muscular dystrophin promoter to see whether it would activate the expression of that promoter, specifically in some cardiac cells. Or you can do it cloning to genetically modified organism. You can clone a protein of interest that will make, let's say, a crop resistant to a certain pest, right? You can do it for multiple reasons. You can do it for doping too, unfortunately. Plasmid have been used for that as well, to genetically modify parts of some athletes' bodies to perform better. So there are multiple applications of what we're going to talk about here. I'll focus on cDNA just for simplicity, but again, the applications are almost endless here. So let's go through a quick overview of the different steps, and then we'll get into the details of DNA cloning. Well, in the very first step, first of all, you have to choose what you want to clone, and then that's going to be referred to as the insert from now on. This could be a cDNA, a promoter, an enhancer, as I said before. So this sequence is somehow extracted or amplified by PCR. And then it's put together, ligated, that's what we say, inside a vector. Okay, here we'll focus on plasmid, but you see in the next slides, these vectors can be of different types. It's put together with the vector, creating this recombinant molecule right here. Now, this is what you're interested on, and this recombinant molecule is inserted inside first bacteria through the process of transformation. Then this bacteria will make more and more copy of these recombinant molecules that will form, first of all, clones, which then you can select by analysis. We'll focus on using restriction enzymes to select which colonies or which clones here do contain the plasmid you want, the recombinant plasmid you want. And then once you figure out which bacteria here, which colony has that plasmid, you would expand it using lots of media here, get it to grow. All these bacteria will be identical to each other, all with the plasmid you want, and then you would extract large quantities of the plasmid. You'll do some of these parts in the lab, okay? We'll do the transformation, for example. We'll do the preparation, the extraction of this plasmid DNA actually very soon in the lab. So this is the overall view here. So again, the insert first is extracted in some way and cloning to the vector. And so these two molecules of DNA are attached to each other, ligated with each other through the process of ligation. Then all the plasmid DNA molecule generated here through the ligation, not all of them would include the insert. There's never 100% efficiency happening. They're all inserted though into bacteria through the process of transformation. And then the bacteria that do have the plasmid are selected, usually using an antibiotics. Then the different clones generated here are analyzed through different techniques, RNA restriction enzyme digestion, PCR, sequencing, and so on. And those that have the plasmid that you want are then amplified, and the plasmid contained inside there is extracted from the clone. So let's start talking about more details of this process. First of all, let's talk about the different type of vectors that you can use in cloning DNA molecule. Here we focus on plasmid DNA molecule, but in reality there are many different type of vectors. In general, these vectors can be divided into several groups. The main ones are cloning vectors and expression vectors. The difference between them is that in cloning vectors, what the DNA molecule that you insert is not going to be expressed. It's not going to be transcribed into an mRNA or RNA molecule. Okay, and these are used just usually to amplify, sometimes just for intermediate steps. Let's say you want to assemble one part of a protein with another part, you can do it through these cloning vectors. You don't need to express these molecules. You just have to cut them so that they can be combined together. That's just an example. You can do libraries with that and so on. Or there are expression vectors where instead, these are great to clone cDNA molecules, so copy of mature RNA are repeated over and over, do not have introns. And so in these expression vectors, there is a promoter that actually can express the RNA, create the mRNA starting from the copy DNA. So what you inserted can be expressed. Now, the vectors, again, here will focus on plasmid DNA molecules, simple ones, very commonly found in labs, but the vectors can be of different types, and all have some advantages and limitations. So plasmid are the circular molecule DNA that are very easy to work with in a lab. They can be amplified very easily, open up, and so on. However, I have the limitations that can only take DNA molecules up to about 10 kilobases, usually much more than that, but up to 10 kilobases. Then you can use viruses, different type of viruses, which can be either specific to human, eukaryotes, and so on, or bacteriophages. And they're a little bit more complicated to produce compared to plasmids. Nothing impossible, but they take a bit longer to generate. However, they have the advantage that you can insert larger molecules of DNA, up to 20 kilobases, now a little bit more as well. And finally, you can use even artificial chromosomes like the yeast artificial chromosomes, which at this point are quite a bit more complicated to generate. However, they have this huge advantage that can take huge molecules of DNA inside, up to 1,000 kilobase, 1 million base pairs. Okay, and also they have the advantage that once you insert them into some cells, eukaryotic cells, they actually do behave like normal chromosomes. So these cells will actually replicate this chromosome and keep them as their own chromosome. So you will permanently modify these cells after you introduce these artificial yeast chromosomes. Similarly, viruses, I forgot to mention that, can actually also permanently, so if you use like retroviruses that do integrate into the genome, you can actually permanently also change genetically modified organisms. But here we'll focus only on plasmid DNA molecules. So here you have some example. Okay, there are many types of plasmid that you can use. So you can search it online. You see there are so many with different names. You'll be working with PBSK, old ones, PBSK and PMBL in the lab. These are old ones, but again, even the PBSK, PMBL have been modified into sub forms. So there are many, many different types. And so the way you choose a plasmid, it ultimately depends on a few things. It depends on how you're planning to clone your molecule. So if you're using restriction enzymes, you'll be limited by the restriction enzyme found in the multiple cloning site. And ultimately, what you want to do you want to express a protein, or you just want to modify it, clone it, subclone it into a plasmid. You don't need to express it. Okay. So in general, though, all the plasmid will have the common properties that we discuss in biochem. Just reminding you, so they all will be circular molecule of DNA. They are extra chromosomal. Okay, so they're not part of the bacterial chromosome. They're naturally found in bacteria. Okay, and they replicate independently from the bacterial chromosome. The reason why they replicate independently from the bacterial chromosomes is because they all have their own origin of replication, which is separate from the one of the bacteria. So all of them are circular. They all have origin of replication, so they can replicate independently. Other important feature, they all will contain one gene which express a protein that confers resistance to an antibiotic to the bacteria that picks them up. Okay, so in this case, all of these plasmid I'm showing you here, they would have a gene that will make the bacterium that picks them up resistant to ampicillin. This is called the selection gene. Finally, all of them that be modified from the natural one by in different ways, and the most important being that they all contain what is called a multiple cloning site, sometimes called polylinker. This is an artificial sequence of DNA that basically contains several restriction sites. So sequences of DNA that can be cut by restriction enzymes. And these have been introduced so that you can easily genetically change or genetically modify these plasmid to contain whatever sequence you want to clone ultimately. Now here, talking about some specific property of these three different types, just to show you the different types you can find. So this will be, there's a common one, PAC19. Later on, I'll talk about PAC18, which is the previous version. Smaller plasmid overall, and this is a cloning plasmid. Why? How do I know? Is because there is no promoter here close to the multiple cloning site here that would allow for the expression of a cDNA cloned here. There is a promoter that allows for the expression of the LAC-Z, but this is a different story there. Okay, that's done for selection. We'll talk about that later on. But there is no promoter here to express anything that is cloned inside here. Other feature that you encounter here is exactly the presence of this LAC-Z, which will help with the selection ultimately of the bacteria that pick up this plasmid. So usually these type of plasmids are used to temporarily clone some sequence of DNA inside, to add other things, or to create cDNA libraries, which were used, for example, in the early microarrays, for example, and stuff like that. The second type here, so both the one in the middle here and the one on the right side here, so PXC histidine here and P target vector here, they're both expression vector in this case. And the reason why is because you can find actually a promoter upstream of the multiple cloning site. So in this case, this promoter here is actually the lac operon promoter. It's a promoter that allows for the expression of whatever it's in clone inside here in bacteria. This would not work in eukaryotic cells, but it will work in bacteria. Together with that, also has an interesting feature that is worth commenting on here, which is the presence of this histidine. This is an open reading frame, which will be connected to the open reading frame of any cDNA you clone inside here, which basically would allow you to express a protein here, which would end with a sequence of histidine. Obviously, all the cloning has to be done so that they are all in frame, so the codons are not disrupted. And so why do you want to have a sequence of histidine at the end is because this will make very easily, your protein very easily purifiable once it's expressed in the bacteria. So if you introduce this plasmid with your own cDNA here, that then produce a protein with a histidine here, then you take the bacteria, you extract the protein that's been expressed from this plasmid, and then you can purify it using these histidine tags. And then you can actually have this cut out at the end. This vector here, finally, instead, it's an expression vector which works in eukaryotic cells. The promoter it has is a CMV promoter, so it's a promoter that comes from this virus, the cytomegalovirus here, which is a virus that normally infects humans. Okay, so if you take human cells and you introduce this plasmid inside human cells with a cDNA cloned downstream here in the multiple cloning site, the protein that is expressed will be expressed constantly through the activity of this promoter. Okay, and so you could see the effect of this protein in the cells that where you have introduced this plasmid. So in this case, this promoter is always active. Okay, so we continuously produce the protein. In this case, I forgot to tell you that this lac operon, and we'll see when we talk about the LAC-Z gene later on, it's inducible. So it's inactive on its own, but it can be induced, okay, by the presence of lactose or synthetic molecules like IPTG. So it would normally be turned off, but if you take the bacteria that have this plasmid and you treat them with lactose or IPTG, they would actually start producing the protein. Another feature of the P-target vector here that I quickly touch on here is these two T overhangs here, which are used in the PCR cloning. You'll see in the next step what they're used for in case you're. So this vector can actually be purchased already opened up with these T overhangs, or it can actually be purchased closed up with this polylinker functional there. So how would you choose the correct plasmid? How do you choose between this plasmid that I just showed you, for example? Well, obviously it's based on the task, first thing. So if you want to express a protein in bacteria, you will use this vector, not the other two. If you want just subclone something, you can use PAC19. Or you want to express it in eukaryotes, you will use a P-target vector or something similar, obviously. But the second thing that it's important to take into account is that now you have chosen, okay, I want to express a cDNA like this one right here in eukaryotic cells. So you get one plasmid that has a promoter that expresses the protein in the eukaryotic cell. Like this one right here as the CMV promoter, like the same plasmid right here. These are different plasmid. PCI neovector, P-target vector here. Which one will be ideal? Well, now at this point, the choice, both of them will work in eukaryotic cells, but the choice is based on your cloning strategies, which is based, if you're using restriction enzymes, on which one has a polylinker which will allow you to clone the cDNA you want to clone, in this example, in the correct orientation. So let's look. Now, these are the restriction sites that you could use to cut the cDNA out and then try to recombine it inside these vectors. Now, there are different restriction sites here that you could use, CLA1, SAL1, XBY1, SMA1. The one you will use obviously will be some that are found also in the polylinker of the plasmid that has to receive the cDNA. Now, however, even though some of these sites can be present in the recipient plasmid, they have also to guarantee that this cDNA is going to be inserted in the correct orientation, right? Transcription has to go, in this case, from left to right, not the opposite way, right? So it means that this cDNA has to be oriented compared to the promoter in this direction from left to right, not the other way around. So which enzyme will work? Let's look at CLA1 is not found in this polylinker nor in this polylinker. So it's out of the picture. You can actually erase it right now. Let me just select the pen for a second, please. So CLA1 is not an issue. Now, SAL1 is present here, and it's present, if I'm correct, in this plasmid here. Great, so it could be used in both plasmid. However, obviously, we're only allowed to cut on this side. Now, what about the other two restriction sites? So X/1 here, it's present in this plasmid and is not present in the other one. However, there is an issue here. If I use SAL1 and X/1 to cut this out and I put it inside here, it will actually be flipped opposite to the promoter. So the promoter would find itself, so if I can use the pen without the promoter, will be on this side. So if you use X/1 and SAL1 in this plasmid, this cDNA will be cloned in the incorrect way. So X/1 could not be used right here. Let's remove this. Now, so let's see SMA1. Can you use it? Well, SMA1 is actually here, present here, together with SAL1, which I've already decided to use. And it's present, so SAL1 here is present on this plasmid as well. But you can see now how, in this case, if you clone the cDNA in PCI neovector, so this vector right here, yes, you end up having the CMV promoter in the correct orientation compared to the cDNA. This will work. In this case, instead, if you look, the restriction sites are flipped compared to the PCI neovector. And so the CMV promoter would end up being on the opposite direction right here, like in the first example. So at this point, you can see you would choose this plasmid right here, which allows you to express in eukaryotic cells and will allow you to clone the cDNA vector in the proper orientation. Okay. Now let's look at the different steps of cloning, okay, of DNA cloning. So first of all, let's talk about the strategies through which you can produce the insert. This can be produced in two ways, by using PCR or by using restriction enzymes. We'll focus mostly on restriction enzymes later on, but PCR can work as well. In fact, it can be produced by PCR, restriction enzyme. PCR has some advantage compared to restriction enzyme. It's actually a faster method to produce your insert. All you have to do is perform a PCR, and you have your amplicon with exactly what you want. It allows you so to control better, if you want, how big the sequence is, where you want it to start, where you want it to end, because you can design the primers wherever you want, pretty much, right, to get them to work. Definitely, it's a faster method compared to restriction enzymes. So in this case, you design your primers, do the PCR what you want to express. You could start from any material. That's another big advantage of PCR. You can start from genomic DNA. Even if nobody has ever cloned that sequence, you can be the first one to clone it. All you need is the primers flanking the region. You're interested on a promoter of a certain gene. Design the primers flanking that region. You make millions of copies of that, and you can clone it. So it's a faster method overall. However, it has some technical challenges. I'll discuss them later on. And as other issues, using PCR, you can actually introduce mistakes. The DNA polymers, they do introduce mistakes. So you might end up having a sequence which is different from the one you want to clone. Using restriction enzymes instead, you'll be using restriction enzymes. So you're limited from a pre-existing sequence, right? You're limited with what restriction sites are present there. So it takes a little bit more planning, but it's very easy to do. in the end. And I'll show you an example later on, but it just takes a bit longer of PCR in terms of steps. Now, both of them can actually be combined, and I'll show you how in the next slide. So if you clone by PCR, well, in this case, as I said before, you could do the PCR starting from the genomic DNA, right? In that case, it's the direct PCR. If, for example, you want to clone a cDNA, all right, so a copy of a messenger RNA, at this point, first, you would reverse transcribe the RNA into DNA, and you can do it with using a poly T primer, right, that binds to the poly A found in all messenger RNA. And then together with that, you can use two specific primers for the mRNA or the cDNA you're interested on to amplify. Simple. Now, if you want to minimize the number of mistakes, you will use an enzyme which has higher fidelity than Taq DNA polymers, like the PFU. So that's the DNA polymers from Paracoccus furious, which has a higher efficiency or higher fidelity than Taq DNA polymers. Now, there are two ways in which your PCR product can be cloned after that. So if you're using actually an enzyme like Taq DNA polymerase that has a lower fidelity, usually you end up with a product that has two very tiny sticky ends consisting of an adenine over N or an A over N right here. So now this can be used, exploited to actually do the cloning. There are plasmid that do end up, like the one I showed you before, with a T over N, which would allow you to clone these as if you were using some sort of restriction enzyme or sticky ends produced by restriction enzymes. However, higher fidelity polymerase, they do not produce this A over N. So in this case, they act more like DNA molecules that are being cut with blunt sticky ends, producing blunt sticky ends. And you could clone them like if you had used that. If you had cut this sequence with with the restriction enzyme producing blunt ends. Now, on top of that, you can actually combine the PCR strategy with the restriction enzyme strategies, as was mentioned before, by integrating on your DNA primers some sequences that do include palindromic sequences recognized by specific restriction enzymes. So in this example here, the primers have been modified with sequences which are not complementary to the original template that would introduce ECOR1 and NOT1 restriction sites. At the beginning, these sequences are not obviously complementary to the template. However, if you remember through PCR, then you start producing amplicons going from here to here. So the more step you do, the more you'll have molecules that at this point would have complementary sequences to these sequences as well. All right. These primers are not super straightforward to use, but they can be easily used. And at this point, you would end up having an amplicon with the ECOR1 site on one end and a NOT1 restriction site on the other end, which then you can use. You would digest it with this enzyme, and you can use to clone it as if you were using restriction enzymes. As I said before, so if you use low fidelity DNA polymerase, like Taq DNA polymers, you end up with these adenine over N. Sorry, there's an H here, forgot, which could be used with some commercially available plasmid that do already have this timing over N. This will help with the clone itself. Or if you use high fidelity one, you would have a PCR product with blunt ends, which can be used with a plasmid that has blunt ends. Could have been opened up with restriction enzyme producing blunt ends or producing different ways. This picture refers to a commercial plasmid produced by Thermo Fisher, but it doesn't really matter. No matter what procedure you use, though, whether you use the A overhang or the blunt cloning procedure, in the end, once you isolate your clones, you will always have to double check by sequencing that the sequence that you get is correct. The reason being that, again, all DNA polymers, whether they are high fidelity or low fidelity, they always have some error rate. And so you can always end up having some nucleotides within the sequence you're cloning, which is not the correct nucleotide. So moving on now and focus mostly on performing cloning using restriction enzyme. Again, we're still focusing on how do you get that insert that then you want to clone into a plasmid. So let's look at how you can do it with restriction enzymes. Just a few words reminding you what restriction enzymes are and how they work. So if you remember, they are endonucleases, so enzymes that cut DNA within the sequence of DNA, not from the ends, and do recognize palindromic sequences, right? Sequences that can be read identically if you read them in the 5 prime. So complementary sequences, they are the same if you read them always in 5 prime to 3 prime, like the one you see there. Now, restriction enzymes will recognize different restriction sites, different palindromic sequences. For example, BIMH1 recognizes this sequence right here. Sorry, I'm going to change color when we talk here. Use a red. Recognize this sequence. Oh man, I wrote the wrong one, and for some reason it doesn't let me write exactly as the one. So it will be this sequence right here for BIMH1. Okay, my pen doesn't work. ECOR1 will recognize this one right here, and so on. Sorry, the pen doesn't work that great. And now, so they all have their own sequence, which you can find easily in any sequence you're working with. And now, once they cut the DNA, they can produce two types of ends. If you remember, some of them do produce blunt ends, so where there is no partial single-strand sequence, like in this case right here. Or some of them produce sticky ends, so partial single-strand sequences, which then can be, they're still complementary to each other, right? Or can be complementary to other structures. Now, both type of enzymes have advantages and disadvantages when it comes to cloning an insert. Now, let's start talking about planning how to cut. So first of all, how do you proceed with cloning restriction site? Well, you have, first of all, to digest both the insert or the plasmid that do contain the insert, for example, you would have to digest it with restriction enzymes, right? So to cut it out, for example, here we had decided, I believe, to cut it with SAL1. This pen is driving me crazy, and SMAL1. Okay, so it's not working. I'll just point easier. So you can cut it with SAL1, for example, and SMAL1. So that's the way you would cut out this insert, for example. And now the vector that is going to accept this insert here, so the recipient plasmid, would have to be cut with the same enzymes or with enzymes that do produce, if you're working with sticky ends, that will produce sticky ends which are complementary to the one created by SAL1 and SMAL1. Now, why do I say that? Because in fact, some restricted enzymes, for example, look at this one right here, they do produce identical sticky ends, even though they recognize different sequences. For example, look at Bagel2 and BAMH1. They're very common restriction enzymes. They do recognize different sequences. Here there is an adenine, here there's a guanine, and obviously the same in the opposite strand. However, in the middle, they have the exact same sequence. So it means that once you open them up, they will produce identical sticky ends. What it means is that if, for example, here there was a bagel one, the sticky end produced on this side could actually be used to clone it into a plasmid that's been opened also with BMH1, right? Because the sticky end will be still complementary with each other. However, obviously, if you do that, once these, let's say, overhang matches this overhang here, the final sequence will not contain neither a Bagel2 nor a BMH1 restriction site, because at this point you would have an A and a G here once this is used. So it's not that you always have to use, normally you will use the same restriction enzymes to cut out the insert and the vector, but sometimes you can use also different restriction enzymes as long as they produce the same overhang. And now you can use blunt ends as well, restriction enzyme that produce blunt ends as well, but sticky ends are usually easier to work, and I'll show you an example why later on. Now, what are the ideal restriction sites and enzyme you want to work with? Well, first of all, the restriction enzymes, ideal ones, they will be cutting only outside of your insert, all right? So for example, in this case, I changed the picture before, CLA1 would not be an ideal restriction enzyme because it would actually also be cutting inside of the sequence you want to get out. If you are trying to clone this cDNA, right, and you digested with CLA1, would cut here and here as well, would not work well. So you want to use SAL1 and XBY1 or SAL1 and SMA1. However, um, you can still overcome this sometimes doing a partial digestion. Basically, you digest your insert in conditions which are suboptimal, so that the restriction enzyme doesn't work at 100%. You get it to work at 5, 10%. So sometimes it might cut here, sometimes it might cut here, and then later on you can actually figure out where the cut took place and still extract this with a CLA1 X by 1 or CLA1 X by 1 strategy. Okay. Now, ideally also, as I said before, you want something that produces sticky ends. They're much easier to work with than blunt ends. In general, they have a higher ligation rate. And compared to blunt ends, also you can more easily, if you use two different enzymes, so two different sites, you can actually control the orientation of the insert, right? I'll show you an example in the next pictures. And then how do you choose the restriction enzymes? It's always helped by using online different tools, which are similar to the one I'll show you later on. So here's just an example how you will work with blunt ends, for example. Imagine this is your plasmid, so that's going to be the recipient plasmid. That's where you want to clone the insert into. And in this case, you open up with this restriction enzyme called HAE3, which produce some blunt ends. Now, when it comes to the insert here, it can be extracted from the original plasmid DNA. So imagine this was the sequence of your plasmid DNA here, the other one, and this is your insert. Now, if you're working with blunt ends, you can work with any restriction enzymes you want. There is no complementarity. There are no sticky ends that have to be complementary. So as long as both of them are blunt, they will work. So imagine you cut this one with DRA1 or DRA1, both ends. Now, what would happen is that once you try to ligate this insert with the plasmid that you have opened up here, now, can you imagine this could actually insert in the proper orientation, the way you want. Imagine that you have a promoter here. That's the way you want it. Or in the opposite way, you have no control in the way you produce. Another problem you have is that when you're trying to ligate, so to combine these things, you would have that your plasmid itself could actually bind with itself much more easily than if you had two different sticky ends on the side. All right. So blunt ends would work. However, they don't allow you to control the orientation very easily, and also they do not prevent the plasmid from easily recombine or reforming the bond with itself. Although there are strategy, dephosphorylator strategy that can help with that. When you work instead with restriction, two different restriction enzymes, you have much more control of how things will proceed. So this will be your plasmid, for example. You open it up with these two restriction enzymes, EQR1 and BMH1, very common one to use. Once they open up, they do produce two overhangs here, two sticky ends, which are not complementary with each other. This already gives you the advantage that this plasmid, if it's fully digested, it will not be able to close up. These are not complementary. They will not bind to each other. Not only that, but now if you extract your insert with the same two restriction enzyme, EQR1 and BMH1, this is how it will look. Now this insert, once it binds to the plasmid, can only insert in one orientation, right? This overhang is only complementary to this one and not this one, and vice versa. This overhang produced by BMH1, this could have been produced by Bagel2, by the way, as well, this same overhang, but here you would have an adenine. This will be complementary only to this one, not the one produced by EcoR1. And so this molecule here can only insert in one orientation. So the advantage of working with restriction enzyme or two restriction enzyme producing sticky hands is that it's much more efficient as a process in terms of preventing the plasmid, the recipient plasmid, to close up on its own without the insert, and it allows you to insert the the molecule that you want to clone in the proper orientation. Now, how do you choose which restriction enzyme to work with? And let's go through an example. Okay, this is something I could ask you to do in a test, a midterm test, for example. So let's go through this example question here. So you're given, what you want to do is you have this cDNA corresponding to the Homo sapiens TIM3 gene. This is a protein that regulates a metalloprotease 3. Doesn't matter. Could be anything. So you have this cDNA, which someone else had already cloned inside this plasmid, called PAC18, the precursor of PAC19 that we talked about earlier on. So it has been cloned inside the multiple cloning site here. This is the multiple cloning site of PAC18, so it will be somewhere inside here. Okay, so it's already been modified. This is the sequence of PAC18. So in blue, you see the sequence corresponding to the multiple cloning site here. And the cDNA will start from here and end right here, okay? Inside the cDNA, you will have also the open reading frame, or ORF here, which is underlined. So that's the sequence that will give origin to the messenger RNA and therefore the protein later on. So this contains a series of uninterrupted codons going from the starting codon here all the way to the stop codon. So now you want to cut this open reading frame, this cDNA, and clone it into a different vector, a vector that would have a eukaryotic promoter. Okay, you want to express the cDNA, you want to transfer it to a different plasmid. How do you do it? So first of all, you need the information about the recipient vector. So this is your recipient vector, pcDNA 3.1 plus, common one. You can use another one, the common one here. This has a CMV promoter. So if you clone it inside here, your cDNA, your TIM3 will be actually expressed if it's cloned in the proper orientation, right? So how do you plan how to cut this out and put it inside there? Obviously, you're limited by the restriction site that you have in these multiple cloning site. Keep in mind that these restriction sites are unique to the multiple cloning site and would not be found anywhere else in the plasmid, obviously. Otherwise, it would be complicated to use them. But in order to do that, so you have to find some of these restriction sites within your sequence right here. How can you do that? You can do that by eye. We used to do it. You can still do it, but it would take time. So if you know the restrictions site for each of these restriction enzymes here, you could look for that. Sorry, surprise room there. Now you could look for them here in all this sequence, but it would take you a long time. Now, luckily, there are online tools like the one you see right here, Restriction Analyzer, which should give me, sorry, let me see if I can get it going here. Yeah, so which you can go. So there are different online tools that allows you to basically to put your sequence inside there and get it analyzed by the tool for the restriction enzymes that are in there. They're all different. Some of them are better than others. The one I used to use was WebCutter, but it's not working anymore, at least the last time I checked. But I love this restriction analyzer tool as well. You have the hyperlink here. So if you want to go there, and which works in a very, very simple way. If you have a good connection here. Sorry, I have Roger. Sometimes works well, sometimes it does not work well. So how does it work? It was working until a few minutes ago, and right now it's not working. So how does it work? There you go. All you have to do in this simple tool here, you have to paste your sequence right here. Now you are familiar with some bioinformatic tools, so you probably know how to use BLAST and so on. It's not too different. All you have to do is put your sequence in what is called a FAST, a very simple format, and then you can get it to analyze it for all the common enzymes, or you can actually provide the list of enzyme right here. What I'm interested on here are these restriction sites. All right, so I'm gonna enter them, the name of these restriction sites. That's all I care about because I have to clone in PCDNA 3.1 plus. And so I already have prepared a document here to save some time. So this is gonna be my sequence. Okay, so we'll copy it, we'll paste it right there. And then I'm gonna add my list right here. Okay, so we'll enter right here. And I have already prepared the list here to save some time instead of enter them. And these are exactly the same restriction site that you find in the polylinker of pCDNA 3.1. Obviously, those that are repeated, like PME1 right here, you just have to enter them once because obviously it's the same restriction enzyme there. So let's paste them there. We save them, and then we'll get this tool to analyze it. The reason why I love this tool is because it gives you a very, very fast way to visualize what you want. First of all, here gives you the list of the enzymes that you enter among the series that you enter that do cut. Okay, so this molecule here, which contains TIM3, can be cut in these positions here by these enzymes. Not only that, but it tells you many times they cut. So these AFL2, ASP781, BMH1, and so on, they cut only once. AP1 and HIM3 will cut twice, and it tells you exactly where it cuts. Same information you get it here summarized, but this is more helpful if you want. Not only that, but also it generates first of all, it allows you to, if you want to produce some sort of experimental gel, how the band will look like, you can do it right there. But also, it gives you a very, very nice way to immediately visualize where these restriction sites are on the sequence. And it will show you those that cut once in bold, so immediately know which one will cut only once, and which one cut two or more times. They're not in bold right here, like HIM3 will cut two times with the numbers right there and so on. So this is a great tool. So now I've already ran this, so we can move on. And I put the same sequence right here. So this is the cDNA, which was in pack 18, analyze with that tool, okay, restriction analyzer. And the only thing that I changed here is I placed the start codon and the end codon of the open reading frame. Why is that relevant? Because this is important to choose your restriction enzymes. You don't want to use a restriction enzyme which is within the open reading frame that you have in there. So which one will work in our cloning and would allow us to put it inside the pCDNA 3.1 multiple cloning site that you can see right here? Well, HIM3 will not work, and I tell you right away, HIM3 cuts before the start codon. That's great. However, it cuts inside as well, so we could not use it. The other one we could use at this end is BMH1. BMH1, which is present here, and obviously is present in this multiple cloning site right here. We knew it because all these sites were chosen based on the multiple cloning site. So BMH1 could work right here. What will work downward? Well, you're limited to equal R1. So we could cut it out with BMH1 and equal R1. So if you take the plasmid, and I'll show you later on, that contains TIM3, you cut it with BMH1 and equal R1, you actually cut out the cDNA with the full open reading frame, plus this additional sequence right here. However, would that work on pCDNA 3+1? Let's see. The CMV promoter is on this end, BMH1 is right here, and eCore1 is right here. So yes, not only we could extract the TIM3 cDNA, but we could actually place it inside this polylinker in the correct orientation. If eCore1 had been before BMH1 here, it would have not worked. So now we have a cloning strategy to extract the TIM3 cDNA for PAC18. So this is a schematic. Obviously, we don't have the exact plasmid anymore here. So what we would do, we take the PAC18 containing TIM3 cDNA, cut out with BMH1 and ECOR1. These would extract the cDNA, right, which will be a little bit more than 863 base pairs because it has some sequences there, and the remaining of the plasmid right there. So if you run this on a gel, that's how it will look. You would have a smaller band here corresponding to the insert you want, and another band, assuming that there is full digestion, obviously, which will be at this level right here, corresponding to the remaining part of the plasmid. Now you can actually physically cut it out. You take a little razor, cut this band out, and purify it out of the gel. So this will allow to have this insert. Now, at the same time, we could take pcDNA 3.1 plus, digest it with the same restriction enzyme, which obviously do not affect the CMV promoter. We'll take out a very short sequence of DNA right there. It's a few base pairs. You would not see it on a gel. But the remaining part of the plasmid, assuming that the digester is at 100%, will look like this. It'll give you this type of band between 5,000 and 6,000 base pairs. You can cut it out and purify it. And then you can mix these two together to try to form your recombinant plasmid with the TIM3 cDNA properly oriented compared to the CMV promoter. We'll take a pause here, and then in the second part, I'm going to discuss how would you proceed after you have cut out these two parts, the insert and the plasmid, and then how would you ligate them together and how would you put them inside the bacteria.